Thank you very much, James and Jason, for the kind invitation. What an interesting and informative conference. And I'm glad to be asked to contribute from a perspective that is a little different from what I've seen so far. So I'm an organic chemist by training, and I run a medicinal chemistry team here at McMaster. These smiling faces here are that team. They're mostly graduate students. We work on chemistry projects and drug discovery projects in a few different therapeutic areas. Most of those areas are not related to cannabinoids or cannabis. We also have some projects in the chemistry of cannabinoids. And that's what I'll talk about today. So a disclosure first, I am actually the founder and majority shareholder of a little company, a spin-out out of McMaster, that develops chemistry technologies that I hope may prove useful one day in the manufacture of minor cannabinoids. This is not actually a great time to be in the cannabis business, as many of you guys know, but nonetheless, that is a company that's related to our research program. And I'd like to start with this bar graph. So we have a tool chemists rely on daily called SciFinder Scholar. It's like a Google Scholar for chemical structures. You can search for compounds and their appearance in patents and papers. And this is the result of a search for the phytocannabinoids. And what you see on the x-axis is the top 25 cannabinoid acronyms based on the number of times that they appear in patent literature, so in patents or published patents, patent applications. So what you see there is that CBD and THC appear in over 3,000 patents, which is a lot for quite a bit for a small molecule. And those patents fall broadly into three categories. The first would be on manufacturing technologies, so genetic plants and extractions and purifications of compounds. The second would be formulations, different additives and methods of administration. And the third, and the area that's of interest to us, is just biomedical uses. When one of these molecules is shown in a preclinical study in an animal or cell model to be potentially related to a disease, and somebody's patented that. So as you can see, after those two, CBG, CBN, CBDV are the acronyms that come up next, and there's a few others that have hundreds of appearances of patents, or that appear in hundreds of patents, which is, for compounds that are not approved drugs, also quite a large presence in the patent literature. These are the 12 most patented cannabinoids. And I'd like today to do a little bit of a tutorial that helps people walk through these acronyms. And at the risk of inducing some anxiety related perhaps to bad experiences in an OChem class for some of you, I gotta say the way to do that is to think about the structures of these compounds. So these are the structures of these 12 cannabinoids. And I've done this before, and I'll be pretty quick. So we're going to start just by focusing on CBD. So on the right is what we refer to as a line angle structure. Every bond, or every kink or corner, represents a carbon atom. The molecule has 21 of these carbon atoms. Those 30 hydrogens that sort of decorate it, we just remove. We know they're there. And then after that, it starts to resemble the line angle structure. Now, let's divide this cannabinoid into three parts. Let's call them face, core, and tail. And then let's just count carbon atoms on each one. So the face will have 10 carbon atoms, the core has 6, and the tail has 5. And the reason I do this is because it makes it pretty convenient to compare between CBD and THC. When you do it this way, you can see that the difference between these two molecules lies entirely in the face. Carbon number 8 is suddenly connected, bound to one of the oxygens in the core. And that is the only difference between CBD and THC. And 3 additional kind of pretty predominant scaffolds and chemical scaffolds in the space are CBG, CBC, and CBN. And as you can see in these guys, the core is also the same. The tail is also the same. The only thing that changes is the face. And in all cases, the face has the same kind of architecture with 10 carbon atoms. So knowing that, I can tell you that if you wanted to understand all of the cannabinoids, you would need to just know 10 phases, and 4 of them are THC. And by far the most dominant THC ones are the delta 9 and the delta 8. And they differ in just the position of the double bond. And then there are 6 others. CBD, CBN, and CBG and CBC are the most common. CBE and CBL are a little less common. There are just 2 cores, one that we call regular or neutral, and one that we call acid. Chemically, we just add this functionality. I've circled in orange there. It's called a carboxylic acid. And when a cannabinoid molecule has that, we put an A at the end of it. And the process of decarboxylation is actually removing that, which happens organically, naturally. When you heat a molecule, the acid falls off, and you lose the A. Right? You don't need an enzyme or a plant for that, just some temperature. And then the tail, cannabinoids have been found all the way from C1 up to C7, but the only ones that are really significant in the plant are the C5 and the C3 cannabinoids. And we call the C5s just regular ones, and the C3 we call varins. The next 2 most common are C4 and C7. So knowing that, hopefully some of you guys would be pretty comfortable if you see THC, also recognizing that a change to the tail is still a cannabinoid, right? Still in the THC variety, we just add a V and call it a varin. A change to the acid, we just add an A, right? And then the CBD family, I did both of those. And if you make a change to the tail and add the acid, you just add both letters, okay? So you call it a CBDVA. Now, I'd like to contrast that with nabalone, okay? Nabalone does not have one of those tails, okay? It does not exist in the plant. And if you're just looking for a linear tail, anybody can just see that nabalone does not have it, okay? Even if you don't recognize that the face of nabalone also has a feature that's not in the plant, that we call that a ketone. That's not natural. And then the other thing I've drawn there doesn't resemble any cannabinoid at all. Okay? If you see a molecule like this, they're still called synthetic cannabinoids. And the reason is because they hit the CB1 receptor really hard, right? And so does nabalone. Nabalone's much more potent agonist of CB1 than THC. And for that reason, these synthetic compounds can be used for the same purpose, right? For the biology associated with agonism of the CB1 receptor, but not any other receptors that THC may target. So that's it. That's the tutorial. Congratulations, you survived the organic chemistry class. Now if I line up all the faces on the y-axis on top, and then both of the cores and 4 of the tail lengths on the left, here's a panel of all the cannabinoids with their acronyms that kind of makes sense now logically, right? All the faces have their own version. So I have delta 8 and delta 9 THC up there. And in this case, the next 10 most patented are highlighted in blue. Now, we're going to leave the chemistry and talk about the biology briefly. And I want to talk about the biology of the minor cannabinoids. But I'm going to start with just the biology of the 2 big ones. And I think from my perspective, by the way, the perspective that I bring to this space is 100% a pharmaceutical researcher, interested in these molecules from the perspective of drug discovery, not at all the recreational use, not the policy space. Just, can they be drugs? Can they help with the disease? I think most people in the room will recognize that this is a very old drug. It was first approved in 1985, and it is 100% THC. Okay? It was first approved by a company called Abbott, it's made synthetically in a lab. It's not extracted from the plant, but the structure of the molecule is THC. Right? Now, here's the FDA label and the indication. I'm sure most of you guys are aware that the FDA approvals for THC are old, 1985, and there are 2 indications: anorexia associated with weight loss in patients suffering from HIV/AIDS. And one of the reasons this was approved, and approved pretty quickly, was the public and policy pressure from the patient community suffering from AIDS in the 80s. And then the other indication is nausea and vomiting associated with cancer chemotherapy. And in this space, this was a Me Too drug that followed nabalone. So there's another company that got nabalone approved in this space first, And then it turns out THC is less potent but does the same thing. So you have to give it in higher doses than that alone. But that indication is nausea and vomiting. The dosages for appetite enhancement are 2.5 milligrams twice daily, 1 hour before lunch, once before dinner. The major side effect was feeling high in some patients and sedation. Patients did not want to take it before breakfast for those reasons. And the dosages for the nausea are 5 milligrams per meter squared. Now, people are not usually measured in meters squared, but they are by cold-hearted pharmaceutical scientists like myself. I'm about 1.9 meters squared. The average man is roughly 1.9. So that would mean about 10 milligrams, 1 to 3 hours prior to taking chemotherapy, and then that 4 or 6 times a day, the same day that you're taking the chemotherapy treatment. So you're certainly going to feel high in the second indication there. And here is from the FDA label, the double-blind, placebo-controlled appetite change data. Okay. So this is a 6-week treatment of this twice a day, 2.5 mg capsule of pure THC. And you see that after 2 weeks, there's really no difference. And this is a appetite measurement score survey. But then after 2 weeks, they start to diverge, and this is why this was approved, right? Now, because this is an old drug, it went off label a long time ago. There are a lot of generic manufacturers. There are 6 of them: Akron, Camber, Lanet, companies you've never heard of, not major pharma companies, just manufacturers of THC. Mylan. It's not available in Canada, but it is available in many countries. It's used a lot in Germany. There's a company out of Texas called Benuvia, who, I don't know how, but in 2013, under the name Insys, they got this reapproved again. And the only thing they did was change to a solution, right? It's just THC. But it's once again reapproved, so now they're the only ones, until their patent runs out, that can sell it in this form. And then I can walk into 1,500 dispensaries in Ontario, and I could just get the same stuff as a candy, right? Which fascinates me about this field, right? There are not any other examples I'm aware of a prescription drug that you can also buy as a candy. Coffee, okay, caffeine. So I don't know, some of you know, caffeine is prescribed to reduce apnea in preterm babies. But that's the only other one that comes to mind. There's probably others. But this is a strange world that we're in here, right? The fact that you can self-administer prescription drugs from these suppliers. And then the other one is Epidiolex. And Epidiolex, they're not trying to hide the name on the label like Marinol is. Yeah, they just say it's cannabidiol, right? It's non-psychoactive. It's a little easier for the public to swallow, I think. It was just approved in 2018. This is its label. The recommended dosage is 2.5 milligrams per kilogram twice a day, so 5 mgs to start, and after a week you move up to 10. Now I have an 8-year-old daughter, and she stepped on the scale at 32 kilos this morning. So that would be 320 milligram dose to a child her size, which is a lot, okay? And if you were to go up to the highest recommended dose, it'd be twice that, so 640 daily. And there is a safety note about hepatic impairment, which means that these dosages, physicians are concerned about liver damage. And depending on that information, the dosage would be reduced or this would be discontinued. Here's the data. So this is placebo over on top versus 10 milligrams per day per kilogram, Epidiolex versus 20. The number of patients next, and then the baseline period, which is the number of seizures per 28 days per month. And those numbers were 80, 87, and 86 seizures. And then down below is how many fewer seizures the child had after Epidiolex treatment. And they fell by 37 and 42 seizures per month compared to placebo of 17. That's a pretty dramatic effect. I am personally shocked that this is not approved in Canada. These are other treatment-resistant seizures. James and I were just talking about the fact that it surprises us that parents are not advocating for approval. Jazz is not interested in the Canadian market. Health Canada does not have a mandate to sort of bring this stuff in. So that's my personal opinion. I'm surprised that this is not available in Canada. And I know some Canadian patients are advised to go to the dispensaries to buy enormous doses of CBD without quality control for keeping the THC out of it. So this entire thing is a little scary to me. But it is helping a lot of children in the US and in Europe. All right, so there's the two drugs, THC and CBD. There's a third one, you may be aware. It's called Sativex or nabiximols. It's 50-50 THC, CBD, and it's approved not in the US, but in the UK, Canada, Germany, other countries. And this is for reducing spasticity in patients with multiple sclerosis. And here are all of the completed phase 3 clinical trials with published results that anybody can look at for these 3 products. Okay? Now, in the case of THC, the data was in the 80s and 70s, and it's not available online for appetite stimulation or for chemotherapy-induced nausea, but there are clinical trials for pain, opioid dependence, and skin picking. CBD has been, so GW Pharma, who pushed this through approval, funded a lot of trials on all of these epileptic indications. And the same company also did trials in Rett syndrome, postoperative pain, Fragile X, and urinary stones. And then this combination, which is also owned by GW Pharma, has also been extensively trialed. And these are phase 3 trials. So these are very expensive, double-blind, placebo-controlled trials. And unfortunately, in these trials, there is no change from placebo or insignificant change from placebo for these highly purified mixtures of these 2 compounds. Which doesn't, just to be clear, it doesn't mean that it doesn't work. It just means that there's insufficient evidence based on the way we conduct trials that it doesn't work, right? So if you are convinced that it works, my advice would be to pay for more trials, as hard as that is to do. Now, GW Pharmaceuticals was sold to Jazz in 2021 for $7.2 billion, kind of at the height of the cannabis bubble. And these are the 2 products that were purchased. Epidiolex is now selling like $1 billion a year. This Sativex is not selling that much. But the deal also included more than 40 patent families that with IP, intellectual property, that's based on preclinical connections between molecules and indications. And here in blue, I've highlighted some of the other molecules that were of interest that are of interest to Jazz, that they purchased IP on for indications. So neuropathic pain and dermatitis and Alzheimer's, and cancers, and another molecule for seizures. So you recognize these acronyms from the tutorial, right? These are not CBD and CBG. These are other small molecules. And if you were to line up the top 12 from before on the y-axis and then just show how many have been evaluated in phase 1, 2, 3 trials, you see it's not many, okay? This is very early research. These compounds also have never been used in large doses by humans because they occur in the cannabis plant in such small doses. So even the perspective of safety has to be all worked out. There are certainly no trials on any minor cannabinoids with published phase 3 results. The closest to that would be CBN. I'm going to go through a couple of these indications for you guys quickly. Not all of them, but in the case of CBN, the registered trials are for insomnia, epidermolysis, pain after surgery, and facial pain and headache. And then in addition to these clinical trials, there's a lot of academic literature that connects some of these molecules to various indications. And it would be impossible to summarize them quickly, but in this case of CBN, just one paper. So ALS delayed the onset of CBN treatment, delayed the onset of abnormalities in a mouse model of ALS. If I look at CBD now, the next one on the list, autism, epilepsy, alopecia, Prader-Wills syndrome. These are all registered clinical trials that are ongoing, no results published. And then there are mouse models, lots of animal models for these cannabinoids. Rett syndrome is a a rare, horrific neurodevelopmental disorder, and CBD improved motor learning, and increased brain weight. As morbid as that sounds, that's what they do after autopsy. They weigh the brains of these animals, and that's a good thing. Here's CBG. Clinical trials registered include sleep in veterans, chronic migraine, appetite stimulation. In the case of migraine and stimulation of appetite, it's with CBD and THC, or with THC, and then ADHD. Also really pretty compelling evidence in mouse models of Huntington's disease as a neuroprotective agent, neuroinflammation in cell-based models, so a long way from the clinic, and a small human pilot study funded by a company called Willow that was trying to find evidence for their product as an anti-inflammatory skin cream ingredient on a small human study. Now, CBG is also an antibiotic, and this is the explanation for why our lab, became involved in this space. This is a paper that we published with my colleague Eric Brown, who researches antibiotics. And it turns out CBG is a pretty potent antibiotic in a mouse model of methicillin-resistant Staph aureus. Now, it's not good enough to ever make it to market in this space, so this is purely an academic exercise. We were wondering about how it works. We answered some of those questions. We wanted to see if it works in a mouse model, and it did. But the unusual part of this, and the reason why we got involved, is because in order to run a mouse experiment, you need like a gram of CBG. And Eric Brown was purchasing these reference standards that cost $250 for a milligram in a mill. So he would have had to spend $240,000 for a gram of CBG. So he just asked me if I could make it. And of course, organic chemists love being asked to make a molecule, right? So without knowing anything about the cannabis space, a few years ago, I was just asked, could you make us a gram of CBG? Didn't even occur to me that it might not be legal at the time. We just make molecules. We're in drug discovery. They're not for human use. So without any concern or issue, we just made a gram of CBG, right? We just found a literature procedure. These are two non-cannabinoid starting materials. One is called geraniol, one's called Livital. They're both available on the bulk chemical market. They're not very expensive. Now, I'm not going to dwell on this, but if somebody would like me to, ask me after the talk, and I'd be happy to. But the nature of a molecule is defined by its chemical structure, not its origin. Okay? So these are the same molecule. It doesn't matter. As long as it's pure CBG, it doesn't matter where it came from. So we made Eric some of the phytocannabinoid CBG, not the synthetic cannabinoid, which in the language in this space, synthetic means non-natural. Okay? So a non-natural cannabinoid can only be made by synthesis, but a natural cannabinoid can be made by the plant or by synthesis. Okay? This is a literature, won't mean much to most of you guys, I imagine, but the actual conditions, chemical conditions required to do this are boron trifluoride in one case, toluene sulfonic acid in another, chloroform. Sounds a little morbid, but we remove all these things when we purify the molecule. And it didn't work as well as advertised, but it was enough for the paper. And now I'll share one slide about how organic chemists think the problems that we encountered and what we actually do. The problem in this case was called site selectivity. So when you combine these molecules, geraniol and Livital, they tend to want to form in two ways. And what we're generating is a bond between two carbon atoms, or a carbon-carbon bond. And the orange carbons on a Livital lead to abnormal CBG, which is structurally different from CBG, behaves very differently. It is not related at all to CBG from a biological perspective. And the other one is normal CBG. And it turns out that inherently, abnormal wants to form, okay, in a lab, in a flask. Now, the enzymes in the cannabis plant do not have this problem, right? They're engineered to chaperone how these molecules come together. You never see any abnormal CBG. In the lab, you do. And this is an analogy that I sometimes use to describe this. Imagine that every molecule has a preferred way of floating, right? So CBG floats in this way, and therefore it can only react on the carbons that are submerged. We discovered a new process that flipped this regiochemistry. We patented it. These are my colleagues that were most influential in this development. And that was the basis for our company. And the way we describe this is to say that we discovered that a substance called alumina, a really common substance, chaperones this reaction and reverses the inherent selectivity. This was industrially valuable for the synthesis of CBG. Bulk manufacture of CBG. is a problem. So the botanical CBG is not pure enough. The next technology is this biotechnology where you take the plant genes and you put them in yeast. In that case, it's a little too pricey. It has not proven to be competitive from a cost perspective. And chemistry was too expensive because of that selectivity problem that I mentioned. And that's an oversimplification, but it is true that that was the problem that needed to be fixed in order to make the chemistry cost competitive. And so our technology is able to make pure CBG. Now, I'll just finish by saying that CBG, our objective now is to make all of these to support preclinical research. If some of them are responsible for some of these activities, it'd be good to know. The world that I play in is not in human trials or even animal trials. It is in pharmacology and enzyme targets. And these cannabinoids are most often associated with CB1 and CB2, but there are a lot of other potential targets on the radar that THC and CBD do not hit, but some of these minors are already well documented to hit. But the data is very incomplete. And the main reason why the data is incomplete is because the compounds are not available in high purity. You can't buy them anywhere. You certainly don't have access to them from collaborators or colleagues. We, I think, in our lab probably already have the world's most diverse supply of minor cannabinoids. And we're happy to share them with anybody doing this kind of research. So I'll finish by reminding you that these 3 drugs, I think it's remarkable that this plant has led to 3 prescription drugs, even if none of them are the sort of chronic pain indication that there's so much anecdotal evidence for. It is still remarkable that 2 compounds from the same plant ended up in prescription medicines. And perhaps some of these other ones are also involved in bioactivities, and we're going to be one of the labs working in this space to try to demonstrate that. And then the last thing that I'd like to leave you with, hopefully, is some recollection of this, especially the cores and the tails, so that when you see the structure of a cannabinoid, you're able to sort of intuitively recognize that it is, in fact, a phytocannabinoid. Thank you for your attention. I'm happy to take questions.